Students, all right, so today we're going to cover the last part of our section 9.2, and we're going to be looking at translocation rate, and we're going to focus on this application, which is going to have us analyze data from experiments. So you don't really have to recreate the experiment or explain the experiment, but you have to be able to analyze data from these experiments. But it's good to know how these experiments are carried out. So if you want to measure how much phloem is being transported throughout the plant, you do so by radioactively labeling carbon dioxide. And we know that carbon dioxide is necessary in photosynthesis for plants, and that carbon dioxide are going, after photosynthesis, carbon dioxide is going to turn into glucose. And then we know that those glucose are going to turn into sucrose, and then we know that this is going to be the phloem in conjunction with a little bit of water that is going to be carried in the plants. So if you want to know how much of the sucrose in the form of phloem is traveling throughout the plant, then you're going to want to radioactively label the carbon dioxide. And you're going to use insects called aphids to measure the rate of that sap that's flowing. So let's start one step at a time here. So what are aphids? Aphids are, here's a picture of the actual insect. They're a group of insects belonging to the order Hemiptera, which feed primarily on sap extracted from phloem. If you look at a picture at a close-up here of the aphid, notice that they have a protruding mouthpiece called a stylet. So it's this little mouthpiece right here, right? And it pierces the plant's seed tubes to allow sap to be extracted. So here you see a diagrammatic representation of the stylet penetrating the stem and reaching all the way to the phloem. Now, the stylet allows this penetration of the seed tube elements because they have these digestive enzymes that soften all these intervening tissue layers. And scientists have tried doing this with a needle of some sort, and they realize that it doesn't work because as soon as they inject the needle and then they try to see if the phloem will flow out, they don't get it to work because these enzymes that are introduced along the way soften all these tissues and then allow the phloem to kind of flow out. So what scientists do, and this sounds a little bit barbaric, but what scientists do is once the aphid penetrates all the way to the phloem, they sever the stylet. So they break the aphid stylet. And then the sap is going to continue to flow from the plant all the way out due to the hydrostatic pressure within the seed tube. So as the sap flows out, there it is, scientists can then measure the rate, right, of this phloem being released from that stem. So how is that measured? So let's look at how the measuring part is done. So we said that scientists use aphids to collect the sap, and they place aphids at various sites along the plant's length, and thus provide a measure of the phloem transport rate. So they start the experiment by growing a plant in the lab within a sealed glass chamber, and they introduce the radioactively containing carbon, and it's carbon-14 that they introduce into the chamber. So the leaves, of course, are going to do photosynthesis. They're going to convert the carbon-14 in the carbon dioxide into radioactively labeled sugars. And then that sugar that is made here in the leaves, it's going to travel from the leaves to the stem, and it's going to reach the first colony and then the second colony and third colony and so on and so on. So once the feeding has commenced, the aphid stylet is cut and severed and the sap continues to flow. So then the scientists need to make sure that the sap that's flowing is the sap that contains the radioactively labeled sugar made through photosynthesis. And then you calculate the time that it takes for that sap to reach this distance, for example. So, for example, if you place your first aphid colony or your first aphid here, 20 centimeters, or actually, you label your starter colony and then you place your colony 1 20 centimeters from the starter colony. Then that is going to be, and then you measure the time that it takes for the sap to travel from the starter colony to colony 1, then that is going to be your rate. So if this is 20 centimeters and it took 1.25 hours for the sap to travel from here to here, then, let me move this up a little bit, then you divide that and you get your rate, which will be 16 centimeters per hour. So as you're going from this colony to this colony to this colony to this colony, you are measuring the distance from one colony to the other. You let the aphids penetrate all the way to the phloem, and then you cut their stylets, and then you see how long it takes for that sap to flow from one colony to the other. And there are some factors that are going to affect the translocation rate, because remember how the sap is moving along here, that's the translocation rate. And what are some of the factors that are going to affect how much phloem is produced and at what rate it moves along the plant? So the rate of photosynthesis is the first factor. Of course, you're not going to get any sap, you're not going to get any sugar without photosynthesis. So we know that photosynthesis is affected by light intensity, carbon dioxide concentration, and temperature. And then the rate of cellular respiration also affects translocation rate, because if the plant here is undergoing a lot of cellular respiration, then it's taking in some of the CO2. And it's taking in some of the oxygen, that is being released. And it's using some of the sugars itself for energy. The rate of transpiration also plays a role, because if some of that water is leaving the leaves, right, then there's not going to be enough water to mix with the sugars and form that phloem, or allow that phloem to flow at a good rate. And then the fourth factor that affects translocation rate is the diameter of the sieve tube. So remember that sieve tubes are a whole bunch of sieve cells that are attached, right, one next to the other. But of course, if one sieve tube happens to be wider than the other, then there's going to be less hydrostatic pressure here. And then that's going to affect how that sap is going to move. So if plant 1 has narrower sieve tube cells than plant 2, right, then there's going to be a higher hydrostatic pressure here in plant 1 than there is in plant 2. And then that's going to affect how the phloem moves. In your activity, and I want to go over this just a little bit, you measured two ways. How phloem was able to move along the plant. So this first diagram here shows how, for example, just one particular part of a leaf was exposed to radioactive carbon-14 or carbon dioxide. So you have to enclose this leaf of this plant. And let's say you expose this one leaf for 3 hours. The autoradiograph image right here, and you can't see it very well, but 6 hours later, you see that the carbon dioxide that got converted into sugars was only able to travel to this part of the plant, right? Because the leaf was situated up here. If the leaf is situated in the middle, notice that the autoradiography shows that the carbon the carbon dioxide that was converted into glucose and sap was able to flow more here towards the top, but also here in the bottom. So if you have those leaves that are concentrated in the middle part of the plant, you see that phloem is moving up and down, whereas the phloem that comes from these leaves that are at the top just stay up here. And if your leaf is right here at the bottom, notice that the phloem that is traveling is mostly staying at the bottom here. So this phloem is making a dip right here and going to its sink, which the sink happens to be the root, right? And not the flower. So this is what this diagram wanted you to show you, that phloem translocates to different parts of the plant depending on where the leaves are and the number of leaves and the rate of photosynthesis and all that. The other picture here in your activity has you looking at the translocation rate via this graph. Let me make this smaller here so you can see. Oops, where'd it go? All right. So on the y-axis, you have the radioactivity in counts per minute, and on the x-axis, you have the distance from the treated leaf. So notice that right away, you see a lot of radioactivity here, very close to the treated leaf that was treated with the carbon-14, right? But notice that those leaves that are farther away, right, they didn't get any of the carbon-14. So this just goes with what I was saying, right, that depending on the leaf, right, like let's go back here, depending on the position of the leaf, the sugar that this leaf makes is only going to travel to a portion of the plant, whatever is closest, right? So that's it for the translocation experiments. Make sure to review them, and we are done with 9.2.